This week, we're going to be focusing on the problem of gastrointestinal diseases found in Chapter 6 of your textbook. Gastrointestinal diseases are a significant problem in the United States, and it certainly warrants that students that are studying in healthcare get some time to focus on the problem. In fact, between 60 to 70 million Americans are affected by gastrointestinal diseases every year. The yearly death toll amounts to a little over 250,000 deaths. There are 10 goals I wish to achieve in this lecture. First, covering constipation, reflux disease, peptic ulcers, diverticular disease, hemorrhoids, anal fistulas, inflammatory bowel disease, irritable bowel syndrome, short bowel syndrome, digestion, and celiac disease. Be sure to complement this video lecture with readings of Chapter 6, for there is far more details in the book as there are in the video. It's in fact pretty hard to dispute the facts that gastrointestinal diseases are significantly affecting American health. Well, it's not really that difficult to understand why it is that many Americans suffer from gastrointestinal diseases. From a broad perspective, we overeat. It's now understood that overeating, lifestyle, and quality nutrition are the driving forces behind many gastrointestinal diseases. As we begin to study these diseases, it's important that we consider the anatomical review of the GI tract. It's important because gastrointestinal diseases and conditions can be better understood if you have a relatively good understanding of how organs are arranged in the peritoneal cavity. The abdominal anatomy that we're looking at is better organized if we set it up in quadrants. In this manner, the physician could describe where the pain is coming from, perhaps the upper right quadrant, the upper left quadrant, the lower right quadrant, or the lower left quadrant. I want to take a look at the highlights of each of these quadrants. If we begin with the upper right quadrant, we can see the gallbladder, we can see part of the liver. We can also see a significant amount of the pancreas. We can see half of the ascending colon, and we can see also half of the transverse colon. Now we can move over to the upper left quadrant. In the second quadrant, we can see that we have the rest of the liver, we have most of the stomach, we have a little bit of the pancreas, and we have the second half of the transverse colon, and we have the first half of the descending colon. Now let's examine the lower right quadrant. So in the lower left quadrant, we see essentially the descending colon. That's the second half, and this is where there is a more drying out of the fecal material. This is where the fecal material is formed and consolidated. And then we have the rectum, and then in the center we have the jejunum, which is the second segment of the small intestine. There is the duodenum, or the duodenum, the jejunum, and finally the ileum. Finally, we can take a look at the lower right quadrant. So in the lower right quadrant, we see the first half of the ascending colon. We see the cecum, and we also see the appendix. Let's do a quick anatomical review of the gastrointestinal tract and the digestive processes. So we begin with the main structures, the oral cavity, the esophagus, stomach, small and large intestine. And then we have the accessory organs, the pancreas, the liver, and the gallbladder. It's assumed that if you're taking this course, you do have an anatomy background. So this will simply be a brief review. So for the sake of having a better understanding of the role of nutrition and how it interacts with the various organs, I'm going to start off with the accessory organs. I want to start off with the oral cavity, which has a mechanical purpose of chewing and breaking down. And on the left-hand side, the salivary glands that provide a mucus environment rich in salivary enzymes that begin the actual digestive process. Now, the pharynx is really important for propelling food that's in the back of the oral cavity down into the esophagus. The key enzyme of interest in the salivary glands is amylase. It begins the digestion of carbohydrates. Well, next, I want to look at both the liver and the esophagus. So anatomically, the role of the esophagus is to bring food from the pharynx all the way to the stomach. The liver's role is primarily to produce bile, which is important in the emulsification of fat in the GI tract. The next set of organs I want to look at are the gallbladder and the stomach. Once food enters the stomach, it is mechanically stirred and it is broken down through chemical processes, notably hydrochloric acid. This is where the proteins are denatured, and ultimately there is the formation of a product called chyme, which then enters the duodenum via the pyloric sphincter. When we're looking at the gallbladder, however, the gallbladder actually, its role is to store bile and to secrete it into the gastrointestinal tract, which then goes through the process of emulsifying the fats that have been consumed. The key concept I want to highlight here is emulsification. To emulsify means to allow the mixing of water and fat together. In this manner, large clumps of fat are allowed to be dispersed into smaller droplets called micelles, which are then more favorably digested by the enzymes. The enzyme in question that is of importance is called the lipase enzyme, and it is secreted by the pancreas. The next set of organs that I want to look at is the pancreas, the small intestine, and the large intestine. The pancreas is responsible first for secreting bicarbonate, which neutralizes the chyme, which is acidic coming from the stomach, and then it produces a set of enzymes for the digestion of carbohydrates, notably amylase, and it also secretes, importantly, lipase enzyme, as previously mentioned. Then the small intestine, consisting of duodenum, the jejunum, and the ileum, is the place of very important enzyme digestion. And it is in the small intestines that a lot of the absorption of key nutrients take place. Now, by contrast, in the large intestine, we're not looking so much at a lot of absorption taking place, but a lot of the material moving from the ileum into the large intestine is actually prepared for evacuation through the rectum. And it's in this segment of the intestine that we see a lot of water reabsorption and also a solidification of the fecal material. So for the pancreas, the key enzymes to remember here is lipase for fat digestion, amylase for carbohydrate digestion. Secreted from the small intestine, we see enzymes for protein like protease, peptidase, and we see enzymes for carbohydrates like sucrase and lactase. Now I want to identify the four main regions of the stomach: the cardia region, the fundus, the body, and finally the antrum or the distal pyloric region. I want to numerically identify these regions for better visibility. So we start with number 1, which is the cardia. And we can see that it's in the region of the lower esophagus as it connects with the stomach. And number 2, we can see is the fundus. This is the upper part of the stomach. Back to number 1, it's actually in the region of the lower esophageal sphincter, which is the muscle that allows the food to move from the esophagus into the stomach. Number 3 is the body of the stomach, and this is where a lot of the digestion is taking place. A lot of the parietal cell secretions are taking place here. Number 4 is the antrum, and this is the lower end of the stomach. And this is where the area is being prepared, where a lot of the chyme, the final product of digestion, is actually taking place. In the body, what we see is a lot of the beginnings of the chyme production through the shaking, the desiccation and breakdown of the proteins, but also the chemical breakdowns with hydrochloric acid. And so this is the region where you have the parietal cells that are secreting both intrinsic factor and also hydrochloric acid. Number 4, the preparation, and number 5, the pyloric sphincter, which is the muscle that releases the chyme into the duodenum. So what you take from this is the stomach is a place of strong peristaltic movement, and it's from here that we also have gastric emptying, right? Gastric emptying through the pyloric sphincter into the duodenum after the formation of chyme is done in the stomach. And chyme is nothing but the breakdown through mechanical agitation and through chemical breakdown of the food products. And this is where we essentially see the denaturation of protein. Once the chyme enters the intestine, it'll encounter large circular folds of the mucosa called folds of Kirchen. The folds are covered with finger-like projections called villi. In between each villi are the crypts of Lieberkühn. And these villi, if we take a close-up right here of one villus, is made up of basically a capillary network that play an important role in circulating the absorbed nutrients throughout the biological system to the various organs that need them. The lacteal is what essentially is called an extension of the lymphatic system. So we have the lymphatic system and we have a capillary network. Now you remember that the lymphatic system was very important in the circulation of chylomicrons, which are really important in encapsulating a lot of the fats, especially the long chain fatty acids and the medium chain fatty acids. What's very important to note is the epithelial cells that are surrounding the villus. All of these epithelial cells form what we call the epithelium. Nutrients broken down and released in the gastrointestinal tract will be absorbed into the epithelial cells. These are nutrients like amino acids, sugars, and fats. And we note that the fats will be encapsulated into chylomicrons, notably some of the medium chain and the long chain fatty acids. And these chylomicrons will circulate from the epithelial cell into the lacteals, which again is part of the lymphatic system, which moves eventually to join the portal circulation, and which will then bring these chylomicrons back to the liver. The vascular capillaries will take the other nutrients and circulate them through the biological system and nourish the various organs that need them. This slide helps students to identify the microvilli relative to the villi. So you can see on the right-hand side the villi, and you can see the projection with the gray arrow indicating the small, tiny little finger-like protrusions that are moving along over the epithelial cells, and these are called microvilli. So it's visible as you look at the right-hand side how the microvilli are protruding out from the epithelial cells, and they're very important in essentially capturing those nutrients that we were talking about earlier. Well, now we're going to take a closer look at the pancreas. And we begin by just simply identifying the pancreas as a set of endocrine cells that secrete hormones into the blood, notably insulin and glucagon. Insulin is instrumental in allowing the body to use glucose, so it allows the entry of glucose into the cell. Glucagon is a hormone that signals the body to actually produce sugar, and it comes into an effect when the sugar levels are low in the blood. The second feature of the pancreas is that it secretes digestive enzymes that are produced by the acinar exocrine cells. These cells will secrete lipase for the digestion of fat, amylase for the digestion of carbohydrates, and trypsin for the digestion of protein. So for clarity's sake, an acinar exocrine cell essentially secretes substance from the pancreas into the GI tract. So it's called an exocrine cell. When we look at the endocrine cells, that basically means that it is secreting inside the biological system. So it doesn't secrete into the intestine, but it secretes into the blood system, and it's called therefore an endocrine system or endocrine cell or endocrine secretions. So exocrine is secreting into the actual GI tract, and this is why it's called. Additionally, the pancreas will secrete pancreatic juices that contain bicarbonate, which were essential for neutralizing the acidity of the chyme that came from the stomach into the duodenum, and it also secretes electrolytes or little minerals. I want to take a closer look at the pancreas, the gallbladder, and the duct system that transports the bile and the pancreatic juices. Well, first, in looking at this particular image, we could see that the common bile duct is a duct that is common to the cystic duct that is secreting the bile from the gallbladder. And it's common also to the hepatic duct, the right and the left, that is secreting bile essentially from the liver. So the liver hepatic ducts join the gallbladder's cystic duct to form the common bile duct. Now this common bile duct merges with the pancreatic duct that secretes the digestive juices from the pancreas, notably amylase, lipase, and trypsin. And they converge together and are secreted, as we'll see, through the sphincter of Oddi into the gastrointestinal tract. So both the bile and the pancreatic juices share that converging point together. In taking a step back and looking at the left image, we can see the cystic duct from which the gallbladder leaves, the bile leaves the gallbladder. And then we see the gallbladder, and then we see the common bile duct, and there where they join together, the sphincter of Oddi. Now, I would also perhaps take a little closer look at the sphincter of Oddi, and you can see the convergence of the pancreatic duct and of the common bile duct here. And of course, they commonly secrete into the duodenum. Now I want to take a look at the cells of the pancreas to clarify what I mentioned previously. There are exocrine cells and there are endocrine cells. The endocrine cells are the cells that secrete into the blood, and they're the alpha cell and the beta cells of the islets of Langerhans. And these secrete, notably the beta cells, insulin, and the alpha cells, glucagon. And these particular hormones get secreted into the blood. Thus, they are called endocrine cells. Now, the acinar cells are secreting the digestive juices, which contain the enzymes amylase, lipase, and trypsin. And these are part of what is the pancreatic duct that joins with the common bile duct to be secreted in the gastrointestinal tract. And these are called exocrine cells. Now, looking at bile and liver more broadly, we can see how the bile gets recirculated to the liver in order to reproduce bile components and also cholesterol. So let's take a closer look. So as a summary, I bring you to number one, and we see that bile is produced by the liver, stored in the gallbladder, and secreted into the gastrointestinal tract to favor an emulsification of fat for easier digestion of fat. We see that the bile is secreted through the cystic duct, and that the cystic duct joins the common bile duct. After the bile has completed its function of emulsification in the fourth step, it recirculates via the ileum back to the liver via the portal circulation in a system called the enterohepatic circulation. These leftover constituents of bile make their way back to the liver, and the liver reutilizes them to resynthesize bile and secrete it back into the gallbladder, and it also uses it to synthesize cholesterol in the liver. This is a closer look at how the enterohepatic circulation actually works. We can see here how the liver produces bile from the right and left hepatic ducts, and how they converge together and align with the cystic duct, which is produced, that drains essentially the bile from the gallbladder, and how it enters the duodenum, moves down the duodenum to the jejunum, and around the terminal ileum, it gets reabsorbed through the enterohepatic circulation back to the liver, where the byproducts of bile are resynthesized into bile again and also into cholesterol. Now, the role of cholestyramine is very specific. It's a cholesterol-lowering drug, and this is how it works. It prevents the reabsorption of bile from the terminal ileum. Therefore, less bile goes back through the enterocirculation back to the liver, and the liver, therefore, is less capable of producing cholesterol. This means that there is a lowering of the synthesis of cholesterol, which means that the liver then relies on bringing in more cholesterol from the circulating cholesterol in the blood, and this lowers the blood cholesterol. So this is how we can summarize the gallbladder. It's made for the storage of bile. It functions to emulsify fat into micelles, smaller little droplets of fat, and that allows for a much better digestion by the lipase enzyme. It's also useful for the recirculation and excretion of bile, and bile circulation through the enterohepatic circulation also leads to, can lead, I should say, to a hypercholesterolemia, because it keeps supplying bile back to the liver, and the liver uses these bile products to synthesize cholesterol, and cholesterol is released. But if you use a drug like cholestyramine, there is a prevention of that cholesterol synthesis, essentially, and that leads to a lowering of the blood cholesterol, as we saw. Now I want to take a closer look at the enzymes that are secreting into the GI tract or into the mouth that are responsible for the breakdown of starches or polysaccharides, complex carbohydrates. And I mentioned earlier, when we were looking at the digestive anatomy, that in the mouth there are salivary glands, and these salivary glands secrete alpha amylase, and they begin the digestive processes of the starches. Then we have the pancreatic amylase, and this is secreted into the intestine. The alpha amylase secreted here will hydrolyze 1,4-glycosidic links in non-resistant starches. So we have amylopectin and amylose. So we saw that organization in Chapter 2. Then we have alpha dextrinase, which is sucrase and isomaltase, and these are found in the brush border um, that basically line the gastrointestinal tract. And these hydrolyze both alpha 1,4 and alpha 1,6, which are the side chains of these polysaccharides found notably in the amylopectin. As we continue to follow the digestion of the carbohydrates, as we previously saw, the polysaccharides are broken down by amylase and dextrinase, and these produce smaller compounds, notably maltose, for instance. And if we throw into there also the sugars that we eat in our food, all of these sort of move towards the microvilli. And the microvilli are the small little finger-like structures that come out from the enterocytes, and the enterocytes are the cells that represent the contour of the villi, if you remember. So we have in this very, very close proximity to the enterocytes the lactase, the sucrase, the maltase, the isomaltase, and the trehalase enzymes that are breaking down these finer sugars and allowing them then to enter the enterocytes, which will then further bring them into the, expel them into circulation afterward. Now I want to look at the digestion of triacylglycerol or triglycerides a little more closely. Well, the first part of the digestive tract that I want to talk about is the mouth, particularly as it pertains to fat, because young babies in particular, because of their immature gastrointestinal tract, actually have a significant amount of lingual lipase right under the tongue that begins the digestion of fat. The chief cells of the stomach also release gastric lipase, and that helps. And the presence of lipids in the stomach have this delaying effect for gastric emptying. It causes a bit of satiety. So the fat that's in the diet gives that sense of well-being in the stomach, a sense of satiety and a sense of being full because the fatty components of the food that's eaten is the last to leave the stomach. Also noteworthy is the digestive fats of short and medium chain fatty acids occur there, and so they are more rapidly absorbed, and this is one of the reasons why athletes will use short and medium chain fatty acids, because the stomach absorbs them quickly, and that actually is looked at as a fast source of energy. Now, in the stomach, there is some emulsification that's going on, and this is happening because of the muscle contractions and also from the squirting of fat through the caloric sphincter. And this emulsification is occurring because of the presence of the polysaccharides, the phospholipids, and some peptic digests, and creates some emulsion there. And then further emulsification takes place with the bile once the chyme hits the intestine. So these bile salts are hydrophilic, and they're also hydrophobic. So the hydrophobic ends are inside, pointed inwards of the micelles, and the hydrophilic part is pointed outside. So the outside part of these micelles that are created attract the water, and the repelling part are in the inside. So the role of colipase is, basically the activation of the lipase enzyme. So colipase activates the pancreatic lipase, and the lipase then continues on with the digestion of the fat. I want to take a moment now just to expand a little bit and review some of what we've learned so far in fat digestion. First thing, in the mouth, especially of babies, we find lingual lipase, and this is because of the the lack of maturity of their gastrointestinal tract, and it's a form of adaptation to help in the digestion of something that would be more difficult for babies to digest, which is fat. And it will hydrolyze short chain fatty acids, or SCFA, and medium chain fatty acids, MCFA, at the third position on the fatty acid chain, and it will release a fatty acid in a 1,2-dihydrogenate diacylglycerol. The bile salts will emulsify the tricylglycerol, or the triglycerides, and this increases access to digestive enzymes like the esterase enzyme to the surface area of the lipids for the breakdown of the fat. And this is, actually taking place within the gastrointestinal tract. And then we have the pancreatic lipase, which so far we've simplified to simply saying lipase. And of course, the lipase is specific to triglycerides, or TG. But we also have phospholipase, which is good for the phospholipids. And we also have cholesterol esterase for the cholesterol esters. Now for the digestion of protein. Now in the stomach, hydrochloric acid, or HCl, denatures protein, causing them to unfold from quaternary to tertiary to secondary structures. So they actually unfold and open up. And the HCl release is stimulated by the gastrin-releasing peptide GRP, acetylcholine, which is a neurotransmitter, and histamine. So all of these work together to get the hydrochloric acid released in the stomach. The peptide bonds of the denatured proteins then become exposed to the action of pepsin. End products are polypeptides, oligopeptides, and amino acids. So polypeptides, complex chains of amino acid. Oligopeptides, a little less long than the polypeptides, and then the amino acids, which are then absorbed into the enterocytes of the villi. Let's look a little broadly and look closely at how the hormones and the digestive processes are linked. So first, gastric cells will release gastrin, which is essential for the release of the gastric juices, notably hydrochloric acid. And again, the gastric juices containing this hydrochloric acid goes on to denature the protein and actually helps in the conversion of pepsinogen to pepsin, which then goes on for the digestion of proteins. The partially digested protein fragments in the intestine will go on to stimulate the secretion of secretin and cholecystokinin. These specific hormones will go on to stimulate the release of bicarbonate into the gastrointestinal tract, notably the duodenum, in order to neutralize the acidic chyme. In addition, it will stimulate the release of proenzymes. Afterwards, the intestinal enzymes and the pancreatic enzymes will go on to further break down the proteins down to amino acids and dipeptides for absorption into the enterocytes. So then in summary, in the small intestine, we have the mucosal endocrine cells that secrete secretin and CCK cholecystokinin that stimulate the secretion of alkaline pancreatic juices, notably bicarbonate that neutralizes the chyme. And also we have digestive proenzymes that become activated in the intestine as well. Notably, trypsinogen becomes trypsin, chymotrypsinogen becomes chymotrypsin, and procarboxypeptidase becomes carboxypeptidase, and proelastase become elastase. These activated enzymes become very important in the digestion of proteins. So when we look at the ending products of digestion in the small intestine, this is what we see. We see dipeptides, tripeptides, and amino acids as the final products that are crossing the microvilli through, or I should say the brush border, into the enterocyte. Uh, Most of this absorption of proteins or of the amino acids and dipeptides and tripeptides are occurring in the upper duodenum. We know that branched chain amino acids are absorbed faster than the smaller amino acids, that peptide absorption is faster than free amino acids, and we know that essential amino acids are absorbed faster than non-essential amino acids. And again, essential amino acids are the ones that are essential for the restructuring of the body's protein system. Greater absorption in the form of peptides, about 67%, compared to 33% from free amino acids. So this is often taken into account by the pharmaceutical company that promote enteral feeds or liquid nutrition, in which they try to increase the proportion of peptides in their formulas compared to just free amino acids. Once the dipeptides and tripeptides make their way into the enterocyte, the cytoplasmic peptidases inside the cell hydrolyze these peptides into free intracellular amino acids, and these are the amino acids that move then into the blood system. So once these amino acids are inside the enterocyte, they must cross through the basolateral membrane, also called the serosal membrane, in order to get right into the blood system. So now let's try a little training quiz to see how well you remember the basic concepts that we just reviewed about digestion. First question, identify the abdominal quadrant in which the descending colon is situated. Stop here and select your answer. The correct answer is C, the upper and lower left abdominal quadrant. Second question, identify the abdominal quadrant in which the stomach is located. Stop here and select your answer. The correct answer is A, the upper left quadrant. Next question, identify the abdominal quadrant in which is located the appendix. The correct answer is D, the lower right abdominal quadrant. Identify the abdominal quadrant in which is located the second half of the ascending colon. Stop here and select your answer. The correct answer is C, the upper right abdominal quadrant. Next question, identify the abdominal quadrant in which is situated the liver. Stop here and select the most appropriate answer. The correct answer is B, the upper left and upper right abdominal quadrants. Next question, place in descending order the segments of the GI tract beginning right after the mouth. Pause here and arrange the series of different segments of the intestine in the right order. This would be the correct alignment: esophagus, stomach, duodenum, jejunum, ileum, ascending colon, transverse colon, and descending colon. Starting after the lower esophageal sphincter, identify in descending order leading to the duodenum the sections of the stomach. Stop here and arrange the sequences correctly. The correct sequence is cardia, fundus, body, antrum, and pyloric sphincter. Next question, where is the capillary network in which the digested macronutrients enter for circulation to the liver and other peripheral tissues? Stop here and select your answer. The correct answer is the villi, C. Next question, where are the crypts of Lieberkühn situated? Pause here and select your answer. The correct answer is B, at the base between each villi. Identify below the digestive organ that secretes bicarbonate. Pause here and select your answer. The correct answer is the pancreas. Next question. Identify below the digestive organ that secretes lipase. Pause here and select your answer. The correct answer is B, the pancreas. Next question. Identify below the digestive organ that secretes amylase. Pause here and select your answer. The correct answer is E, the pancreas. Next question. The common bile duct empties into the duodenum via which orifice? Pause here and select your answer. The correct answer is B, the sphincter of Oddi. Next question. Identify below which part of the pancreas secretes insulin. Pause here and select your answer. The correct answer, C, the endocrine ductless cells. Next question. Identify below which cells of the pancreas secrete glucagon. Pause here, select your answer. The correct answer is D, alpha cells in the islets of Langerhans.